You've seen like the cars with the snorkels, right?
No.
It's like a thing you can install so that if your car starts to get into too deep of water for it to handle, the air intake is above the engine
Oh my
instead
God.
of within the engine.
That's,
So the car can keep running while it's a snorkel. It's literally a snorkel.
oh my God.
From ScienceSortOf.com, you're listening to Science Sort of.
Hello and welcome to Science Sort of. You're listening to episode 335. Our theme this week is Major Herp-o-compatibility Complex. I am your host, Ryan, and joining me to talk about things that are science, things that are sort of science, and things that wish they were science is someone who's herp-o-compatible herself, Kelly Wintersmith.
Hey, everyone. Good to be back.
Good to have you, Kelly. It's been a minute, but it's always great when you're on the show.
Life's been crazy.
Sure has.
Yeah.
Speaking of life being crazy for folks, we're also joined by Gina Zwicky.
Hi. Thank you guys so much for having me.
We are delighted to have you here. So Gina is a New Orleans-based biologist, naturalist, and wildlife photographer, aka the Frog Girl. She studies reptile immunogenetics and leads the New Orleans, Louisiana Frog Watch, or NOLA Frog Watch trips. And she encourages you to go outside. But not right now. Listen to the podcast first, then go outside. So Gina,
or you
thanks
can
for
walk
being
while
here.
Oh
listening.
yeah.
That's true.
That's what I did to prep, so.
I have been criticized on hikes before for having one headphone in, listening to a podcast. They're like, Ryan, we're on a hike. You should be enjoying nature. I'm like, yeah, but my brain gets bored. I gotta put something on.
I feel you. You gotta do the brain hemispheres thing like dolphins, you know?
Exactly. I sleep one half of my brain at a time. Yes.
Epic.
But we're not talking about dolphins. We're talking about reptiles, herps. Question we like to ask folks at the beginning of an interview with them is sort of, what kind of scientist do you identify as? And you can answer that for whatever context or for whatever audience you have answers with which to give.
So typically, if I was explaining my work to just somebody who wasn't in the sciences, you know, like a friend, family member, I would call myself an ecologist. So meaning that more broadly, I study the interactions of organisms with other organisms in their environment. But talking to someone else in my field, I think I might get a little bit more specific and describe myself as a molecular biologist, since the nature of the work that I actually do, the nuts and bolts of it, is working with the genome of the lizards that I study. So I can get a little bit more into specifically what that is a little bit later in the podcast, but ecologist in general and molecular biologist if I'm at, my advisor meeting.
And can we hear more about the lizards?
Yes. Oh my God. Yeah. Oh no, we're very gung-ho about lizards. I really love these lizards. They're so cute. It's a species called Anolis sabanis, or the Saban anole. And they're interesting for a variety of reasons, but primarily because they're an endemic species to a really small island in the Lesser Antilles called Saba. It's about 13 square kilometers in area, so really, really tiny. And endemic meaning this is the only place in the world that they're found. So the fact that they're an endemic species and that they live on an island where there aren't very many other native reptiles that sort of occupy that same ecological niche can make it a lot easier to use them as a system to answer ecological questions. So they're also really cute. Oh my God, they're so cute. So anoles are fairly small. Most of the ones that I encounter are about like 3 to 6 inches long, including tail. These guys about in that range, but they are also sexually dimorphic with coloration. So the females are sort of drab, still fierce, but the males are really dazzling. They have like a beautiful dappled spotted pattern like little leopards. I basically just work with a bunch of dots of their blood that's been collected over a span of over 15 years. So I have some pretty old samples, and I extract DNA from their blood to answer these sort of evolutionary questions I'm working on.
And what sort of makes anoles unique amongst lizards? Like, as a group, what makes anoles?
So anoles are actually more closely related to iguanas than geckos, which not a lot of people actually know outside of the field, just because, they're sort of small, kind of registers in the brain in sort of like a gecko way, if that makes any sense. But another really interesting thing about anoles is that they are one of the classic examples of what's called an adaptive radiation. So the Darwin's finches are another example, an adaptive radiation being a group of closely related organisms that have specialized upon habitats in a geographically contiguous area but all of them are sort of just like a little bit different from each other. So the Darwin's finches have, the slightly different beak shapes that allow them to specialize on different foods. With anoles, which have several hundred species across South, Central, and into North America, it's more differences of habitat use, body size, coloration, etc. So adaptive radiations are fascinating systems in which to study evolutionary questions as well. So I've sort of got like a twofer on just my study species being awesome. So I'm very excited about that.
Now, anoles are awesome, but parasites are maybe more awesome. So importantly, what parasites can you find in the anoles?
Yes. Okay. So wild reptiles are pretty gross. They have a lot of parasites, both ecto and endoparasites. Ectoparasites being stuff like mites, ticks that attach to the outside of the body and usually feed on blood. So hematophagus, there's another 10-point word. And then there's also endoparasites. So that's stuff like...
Wait, you can't just throw out hematophagus
Yeah,
and
right?
not define it. Absolutely not on this show.
All right, fair enough. So hematophagus, so hema, the prefix meaning blood, and phagus, suffix meaning eater. So you can sort of puzzle out a lot of these scientific and ecological terms and figure out what they mean, but this one's very goth, so I like it a lot. But yeah.
My mom used the term vascular necrosis the other day at dinner, a family conversation. and then she was like, wait, do you know what that means? I was like, of course I know what that means, Mom. It's in the word. And then she was like, well, I didn't know if Julie, my wife, would know what it means. And Julie's like, yeah, it's in the word. And we're both just like such science nerds that you can throw out avascular necrosis. And we're like, yeah, we know exactly what that means.
Yeah, like a blessing and a curse right at the dinner table, I imagine.
Yeah.
Is the curse talking about necroscopies at the dinner? Anyway, yes, I actually did a segment on the prefix hema in a previous episode of the show. So people, if they've been listening through the back catalog in order, they've heard me cover that. But now we have phagus to add on to that as a cool suffix.
Yeah, stick around for the quiz after the episode, right? So, but yeah, the endoparasites as well are a major component of the system, and that's actually what I study. So endoparasites, meaning they live inside the body, and specifically what I work on is malaria, which is really common and widespread across bird and reptile taxa. So
is it symptomatic? Or do they get sick, or are they asymptomatic carriers of malaria?
So that's an interesting question because there's a fairly broad range of symptoms that are associated with malaria. There's both an acute and a chronic infection. So acute meaning that it comes on really quickly, has more severe symptoms. Chronic meaning that it's sort of just a background infection that they harbor. But some of the symptoms that you can see in the more severe infections are like lethargy, weight loss, lack of like thermoregulation behavior, stuff like that. So they kind of just
Sounds
look like,
2020.
Yeah, right. Oh my God, like same.
Yeah.
But they sort of languish. You can kind of just tell that they visibly like don't look well.
I remember having my mind totally blown when I found out that birds and lizards got malaria also. I guess I'm like super human centric. And when I found out they had it too, I was like, wow, that's wild. But so that seems like a hard thing to study. One, because you have to draw blood, presumably. And two, because like they all kind of look the same under a lots of blood parasites look the same to me under a microscope. How hard is it to study this?
Okay, so that's actually an awesome segue into one of the cool parts of this project. So we don't actually have to visually identify them. So a really cool thing, Plasmodium infections, so Plasmodium, the genus that these parasites are in, used to be most commonly done by a blood smear. So basically you would take a little drop of blood, spread it very, very thin across a plane of glass, and then check under a microscope, and you would try to do your best to identify the species that you're seeing. But it's not very easy to tell. So there was a lot of cryptic diversity in this parasitic group. So what we do now is we use a PCR test. So polymerase chain reaction. We use primers that are specific to these parasites, the malaria. And then we can actually detect whether or not a lizard has malaria just by testing a drop of its blood with this PCR test. So we don't actually have to do the blood smears anymore. You can do like parasitic load if you want to in that way, but this is a pretty easy, relatively inexpensive and reliable way to determine malarial infection status. And then we take that information and we send off these little pieces of DNA that we've amplified of the Plasmodium that are in this blood sample, and we can send them off to a sequencing company, take them back, and run them against a database. So in this case, the NCBI database, which I'm sure you guys are probably familiar with, but the host or the listeners may not be.
I am not familiar, so.
Okay, so sorry for assuming. But yeah. The NCBI database, which shamefully I cannot remember what it stands for off the top of my head right now, but it's a massive, massive, massive online repository of genetic sequences. So if you get back like a little piece of mystery DNA, you can put it into this database and it'll come back with a variety of, statistical confidence results for what they think it is. And it's crazy how small a piece of DNA you can do this with. Like some of the sequences we're getting back are like 30 to 40 base pairs. So like base pairs meaning A, C, T, G, etc. And it can tell me very reliably, all right, this is Plasmodium floridens or Plasmodium falciparum. And I'm like, oh, dang. All right.
So
we can
is
tell.
That like, is it generally that reliable, or are the malarias so variable that a 30 stretch sequence tells you, but maybe that wouldn't work for another species?
So that's a really good question. So the reliability of kind of finding out what the species is by using the NCBI BLAST function is what it's called. It's an acronym. It depends on how closely related the species are. So most identification of species that you don't know what it is and you want to use their DNA to find out the species is in the cytochrome B gene in animals. So the cytochrome B is part of the mitochondrial genome. So it evolves really quickly. It's a really small section of DNA compared to the larger, broader genome. So even species that are fairly closely related are likely to have some divergence in the cytochrome B gene just because the mitochondrial genome is pretty quickly evolving. It usually is pretty reliable if you're testing like reasonably distantly related species, but for stuff like very recently diverged species, it can be kind of muddy to tell if you're actually looking at different species or if it's just, kind of a fluke.
Ah.
And is it also dependent somewhat on like who's submitted DNA to this database? Like, is it user generated data being input into it? So like things that are more likely to be heavily studied maybe have better confidence intervals?
That is definitely the case, yeah. And it's really useful specifically for malaria because it's primarily a medical database. So there is all kinds of DNA in there for any sort of animal you can imagine, but we have really high quality and very numerous sequences for these malarial genotypes. So I'm pretty lucky in that. I'm sure people working on, obscure new fishes and stuff like that are kind of banging their heads against the wall trying to do this same work, but it is really interesting and it's a great resource.
I was trying to identify the parasitoids that infect the crypt gall wasp, and it was like, I don't know, bug. So
yeah, good luck.
Yeah. So there's a lot of variability in how precise you can get, I think, when you blast.
Mm-hmm. And I think that I'm also really lucky because I work with blood extract DNA because the rest of my lab works with fecal extract DNA, which is relatively very low quality. So there are a lot of missing bases, stuff like that. You have to fill in a lot of the holes to get any sort of usable information. But blood samples often come back very high quality, very pretty. So I'm definitely a little spoiled. I can't complain too much about the quality of my DNA.
And do you get to do field work? Do you get to go to the Caribbean and catch anoles and take little blood droplets from them?
I was supposed to, but COVID. It's so sad. I've done a lot of field work in my time and I get to do a lot of just being and goofing around in nature here. But I'm actually really fortunate that this data set exists. It was collected over almost 2 decades by Susan Perkins, who was formerly at the American Museum of Natural History. Right? Isn't she cool?
I love Susan!
Yeah, so I'm working on her data set. She's also, incidentally, the one who told me that I was pronouncing Saban anole wrong. Oh my God, the most mortifying experience of my life in this meeting. Yeah, I'd been calling it the Saban anole for year.
Isn't Saban Entertainment they make the Power Rangers?
Yeah, but there's also Saba. It's like an Indonesian island. So there's 2 islands that are Saba, and the Caribbean one is Saba, Indonesian is Saba. So I will never forget that.
I'm pretty sure there's a Power Rangers character also named Saba, if I'm being totally honest.
All right, that makes me feel a lot better. But yes.
I think that was the White Ranger's sword. Wasn't that named Saba?
Yeah, it's ringing a bell. So I'm going to cling to that to maintain my pride. Because I'm so embarrassed. She was so nice about it, too. She's so cool. But yeah, so she had these blood dots, and she's done extensive work in Saba. So long time. She's an expert on malaria and host parasite coevolution. And my PI, Nicola Anthony at the University of New Orleans, and her have a long-standing working relationship. So that's how I sort of got onto this project.
Awesome. And so what do you do with those samples then?
So what I'm doing is basically trying to test the mechanism by which variability in a particular segment of what's called the major histocompatibility complex is maintained. And that's a pretty crunchy sentence. So let me break that down a little bit. The major histocompatibility complex is a hypervariable family of genes that regulate immune responses in vertebrates. It's, I think, the most variable gene family in the vertebrate genome. So, it begs the question, why is it so variable? And one of the low-hanging fruit answers is that host-parasite coevolution maintains this variability, but there are several different mechanisms by which this could happen. We think that they're probably all working kind of continuously, and they're not mutually exclusive, but I can go through these main hypotheses just very quickly. One of them is what's called rare allele advantage, meaning that basically it's more likely that these parasites will not have adapted to sort of overcome immune genes that are less common. So it's just as simple, well, if they haven't seen it before or they're less likely to have seen it before, it'll probably be a better genotype to have. And then we've also got the heterozygote advantage hypothesis. This basically just says that individuals that are heterozygous at this locus that we're looking at, so having more than one allele, are able to respond to a greater range of pathogen impacts than something that has less genetic variation at that locus. This is still so crunchy. I'm sorry. Trying my best to make this palatable. But what we're most interested in is seeing if we can use this natural system to test what's called the fluctuating selection hypothesis, which basically says that spatial and temporal differences in the abundance of pathogens and level of pressure that we're getting from these pathogens might maintain diversity at the MHC, because it means that different genotypes will be more beneficial in different times and places. So all of these genes will sort of just be floating around in the ether waiting for their chance to be useful. This is pretty hard to test because there's just so many variables that you need to consider in a natural system. So that's where this data set comes in. We've got 15 years of blood data from an island endemic species. So not a lot of other reptiles on this island to sort of influence transmission patterns, because, you can get habitat sharing reptiles transmitting malaria to each other just through proximity. Mosquito feeds on one, then the other. So we've got that going for us controlled. We've also got no gene flow to this island from other anoles, because there aren't any other ones.
Yeah, and if you look it up on a map, it's actually it's further out on its own than a lot of the islands in the Caribbean island chain. So that was kind of interesting.
Yeah, so it's like way out there. So it's like a little experimental island.
I know paleontologists who have done work in the Caribbean islands because, like you said, they are these sort of naturally occurring experiments. Because in the Pleistocene, when we had these giant ice sheets covering huge parts of continental North America, or what would become North America and the Northern Hemisphere, sea level was much lower. So a lot of these islands were connected. And then we have a moment, 11, 10,000 years ago when the last ice age ended, and these islands became isolated and have been isolated ever since. And so there's a lot of cool work you can do given those circumstances. And this is clearly one of the options.
Yes, very much. So island biology is just fascinating. So many questions that you can just easily test. But in a better synthesized way, what I'm doing is testing these anoles, the blood samples from malaria, and simultaneously on the same individuals. So I will use the same blood dot for multiple things. I'll also sequence for the major histocompatibility complex genome at a specific locus. So for each lizard in my data set, I've got whether or not it has malaria, and then the raw genetic code at this MHC locus. So we're going to see if parasite pressure differs at my different study sites. So there are several across the island and across time, because I have 3 time periods that I'm working with, and to see if the differences in parasite pressure have any sort of correlation with the frequency of the alleles that we're seeing at these loci. So basically testing the fluctuating selection hypothesis directly in a natural setting, which can be a pretty powerful provision of data to support this hypothesis if we're going to find anything interesting there.
Is the malaria fluctuating over time because of something like, the weather wasn't good for mosquitoes one year, or is it fluctuating over time because the immune systems of the lizards caught up? Or can we tease that stuff out?
I think you can. I think that a lot of this you could probably infer with pretty high fidelity. Transmission dynamics of parasites like malaria can be influenced by all sorts of things. So like you said, exactly, bad year for mosquitoes probably means bad year for malaria. There are temperature associations with levels of parasite pressure, but that's not really the question we're asking. It's sort of, adjacent to it. But yeah, it would be really interesting to tease out some of why these levels of parasite prevalence might be differing. But yeah, like you said, any kind of environmental factor could influence that.
And so this fluctuating selection, I actually wasn't super familiar with it. And so, I tend to think of runaway natural selection happening, which often is a sexually selected trait, but that just kind of goes crazy. So if you think about like male peacock tail feathers, right?
Mm-hmm.
And I don't know if that's exactly the red queen hypothesis, because I always thought of the red queen hypothesis as more of like a predator-prey dynamic. But like you have these moments in evolution where something keeps getting pushed and pushed and pushed to a more and more extreme level, to the point where peacocks can barely fly and are very obvious to predators, male peacocks again. But it seems like the idea of fluctuating selection is that like you're able to evolve a trait that's extreme enough to keep you ahead of the malaria, but then it's able to reverse and go the other direction instead of just continuing off indefinitely in one direction. Am I sort of understanding that right?
So you're bringing up something interesting with the red queen hypothesis. So for anybody who's not familiar, this is an oft-quoted hypothesis that basically says it takes all the running that you can do just to stay in the same place. So it's a line from Alice in Wonderland from the red queen, basically meaning that predator and prey and hosts and parasites are basically continuing to evolve over time indefinitely to be better predators, more evasive prey, and more defensive hosts and better parasites. So that's how we get some of these crazy adaptations that we'll see, like anti-predator adaptations in nature, like crazy stuff like the bombardier beetle, like shooting off acid and whatever. But
yeah, one of my favorites is the pronghorn antelope in the western parts of the United States, which is not an antelope. Its closest living relative is actually the giraffe and the okapi, and they're the fastest land animal in North America. They're just way ahead in terms of speed of any predator that currently exists on the continent. So you have to go back 10,000 years again when there were cheetahs in North America. There was an American cheetah, and like suddenly, the reason that this pronghorn is running at 60 miles per hour and eating the feces of its young, it all makes sense, because it doesn't know that there's not cheetahs still out there. It doesn't realize that this predator that it's been evolving alongside for its entire history has gone extinct. And so it's now over adapted for the environment thanks to this kind of red queen hypothesis.
So for the application of the red queen hypothesis in this system, it basically is favoring just having lots and lots and lots of different alleles. So it's not favoring any one specific one, because that would sort of nullify the point of having one particular allele, because if it's just one that was at one point really advantageous, then the parasite is just going to co-evolve to beat that one, and then you're SOL, because you have so little genetic variation to respond to this pressure. So you also got onto something that's, oh my god, I'm so obsessed with like ghost traits and like Pleistocene stuff in North America. This is like totally off topic, but it's so cool, like the giant spines on the trees and the giant sloth. Anyway, I love it.
Oh yeah, Kelly knows not to get me started on sloth, so we're not going to go down that sloth hole.
But it's always interesting. So then I guess
Sloths
what
used to make holes, Kelly. It's true, they burrowed.
Okay, calm down. It's okay, Ryan.
Paleo burrows in the Pleistocenes of Bolivia. I'm done, I promise.
I'm not sure I believe you.
I swear.
I almost got you started too. I was reading something like a couple months ago about like sloth burrows and how they're like an unintentional source of like paleontological samples or something.
I have nothing to say on the matter.
Nothing? Okay, yeah, never mind. Canceled.
I won't. No, I mean, we will literally have you back on the show, and I will talk at you about sloths instead of you talking at me about anoles, and we'll just do this in reverse.
Oh, hell yeah, all day.
And we'll make Kelly sit in on it again, but I won't do it this time.
She can just like sigh.
I enjoy the tangents. I just sort of always feel like it's my job to sort of bring things back on track.
Yeah, you need a straight man in any good group.
Yeah, well, that's my job in my house, and then I just sort of tend to do that in other parts of my life as well. But okay, so the fluctuating part, I guess is what's fluctuating the frequency of the alleles over time, or the frequency of malaria? I guess I'm not quite following what is fluctuating.
So both of them are, but it's the fluctuation in the prevalence of malaria that drives the subsequent fluctuation in these allele frequencies. So, it's a response to different levels of parasite pressure in different time and space factors.
Got it.
That wasn't very eloquent, but I think you're getting me.
No, I'm getting it. And so how does the variability stay maintained then? Because like if you get malaria and there's an allele that is better at maintaining, I know you said it defeats the purpose if you lose your variability, but how does that variability get maintained when selection is favoring certain alleles when malaria is less good at attacking individuals with that allele?
So there's a lot that we don't know about this sort of dynamic between the host and the parasite. And the scope of my project is less so than, the theoretical stuff you're getting into. I don't want to, talk on things that I can't really explain confidently. So
fair enough. Yeah. Okay.
Foot removed from my mouth. It's fine.
And so you're still waiting on results. So I understand if you don't have like any conclusion to this story, but A, I hope you will come back and tell us when you do have some interesting and exciting results to talk about. And B, sort of at this point in your research journey, if a person who listened to this conversation, if you wanted them to walk away with sort of one thing that they understand about what you're trying to do, what would that one thing be?
Sure. So if I wanted people listening to take away one thing, it's just that it's a really kind of complex and beautiful relationship between hosts and parasites, and that there are a lot of mysteries waiting there at the molecular level for us to uncover.
That's awesome.
Job security.
That's super
exactly right.
I love that.
Yeah, we want there to be more stuff, so I'll be employed for longer. It's great.
Right. There's plenty of other islands with plenty of other anoles for you to go get to work on.
Mm-hmm.
Speaking of islands in the Caribbean, I learned today on Twitter from someone I believe is a mutual follower for at least Kelly and I, Andrew Thaler.
Yeah.
He just announced that, I think I need to follow up on what exactly his role is, but he announced that a distillery in Dominica has broken ground, and it is a rum distillery / sea turtle conservation project.
Oh, great.
And I hadn't seen that. Yeah, tell me about that.
That's, as much as I know at this point, other than I want to go on a field trip. But since I can't yet go to this exciting rum distillery / sea turtle conservation hub, I instead will transition us to our segment where we talk about what we're drinking, even if it's not cool Dominican rums. In our upcoming segment, what are we drinking? If we didn't also talk about what we are drinking. Gina, as our guest, would you like to kick us off on the drinks segment?
I would love to. I'm going to share something sort of embarrassing about myself. I have like a 14-a-day LaCroix habit. This is so shameful. Wow. Like my recycling just littered with the ghosts of my sins. But I'm currently drinking a delicious lime-flavored LaCroix. I'm gesturing to it. You can just imagine this. And thankfully, because we have had like unreliable power due to just crazy stuff going on in the South for the past couple days, it's almost like it's refrigerated. It was sitting by the window. So nice. Crisp, refreshing LaCroix, as we say them in my house. So that is what I am drinking right now.
Can you rank your top 3 LaCroix? Kelly, you're a little older than us. Do you consider yourself a millennial?
I don't know.
Okay. So
that's a long pause.
Sorry, I really
I think I'm I know that Julie and I are older millennials, but we have definitely bought into the LaCroix habit as well. So, Gina, can you give us the top 3 flavor rankings?
Oh my God. Okay. Pamplemousse has got to be, number one. Definitely. Lime comes in most of the mix packs, so you got to love it one way or another. It's just inoffensive, It doesn't taste like that much, but what does of LaCroix? So pamplemousse, lime, and then Raz Cran. Those are probably the 3 best ones. The only one that I can't drink because it tastes like sunscreen is the coconut one. That's
coconut
nasty.
One? Yep.
It's so gross.
There's like a limoncello one that is a little off as well.
Oh yeah.
But there's also a key lime if you want to vary up your lime.
Oh yeah, I got both of those at Big Lots, and you're right that the limoncello one was nasty, but the key lime is a good mixer as well. I'll drop that in. But
I would say the 3 on rotation in our household are the pamplemousse, the passion fruit, and the tangerine.
So on a separate note about seltzer, just for the connoisseurs who are present here, Costco does a really good LaCroix knockoff. But if you get them, They explode when you open them every single time. Like, I don't know what's going on with it, but I have to like go into the bathroom or like the kitchen to open it over the sink because it just like spews seltzer everywhere. So very cost efficient, but a little bit, like adrenaline spiking as well. If you're looking for like a spicy seltzer experience.
Yeah, I was doing a Zoom hangout a couple of months ago, and it's a group of friends that are all like, all right, so Kelly's gonna laugh. They are bigger cocktail snobs than me.
What?
They exist. So I forgot to prepare. I didn't think to. They all do like tiki drinks too, which are very complicated to make properly and take a lot of ingredients and a lot of skill, which I don't have. So I forgot to make anything, and I literally had a newly invented cocktail that I named the Millennial Margarita, which was just a shot of tequila and a lime LaCroix.
All right, I call that the same thing, but with like a Baja Blast.
Yeah, there you go. And then there's also the what's the one with the grapefruit and tequila that I'm blanking on the name of. A Paloma. The Millennial Paloma was a pamplemousse with a shot of mezcal. So those are the cocktails I invented that night.
If I'm just exposing myself, like in this this segment for right here, my friends think I'm so weird because I'll just drink like LaCroix and vodka at the function. Like it's economical. It tastes all right. Good enough. So I need more friends who are good at making cocktails so that I won't have to do that anymore.
Yeah, for sure. It's always good to have cocktail friends. Kelly, what are you enjoying this evening?
Well, as the old member of the group, I am just drinking chamomile tea. I've been drinking a lot of red wine lately, but I feel like for a little while I should just switch back to tea. So I'm just having a nice, calm cup of chamomile tea. I know, that's it. I'm just very boring.
That sounds wonderful, honestly. I'm like relaxed just hearing you talk about it.
I've only ever had chamomile tea in like blends. I've never had just like a pure cup of chamomile tea.
Oh, we bought it in bulk on Amazon, and we just have a little like teapot, and we steep it straight up. We got loads of it here.
I got Julie a Appalachian mint tea blend for Valentine's Day that has some chamomile in it.
Nice. We've convinced her
and enjoying it.
We've excellent. We've convinced our oldest that when she drinks it, it makes her sleepy. And that is a useful lie to be able to tell a 6-year-old. So I have a bit, and then she has some, and then she, placebo effect, thinks she's getting sleepy and goes upstairs, and it's lovely. We love chamomile.
Brilliant.
Oh, that's great.
Yeah.
That's fantastic.
Yep. And then she goes up and reads comic books, which is super cute.
What's she reading?
She's reading Captain Underpants and Dogman, and then the Science comic series.
Oh yeah, those are great.
Yeah, they are great.
We have the one on bats.
Oh, we've read the one on bats and dinosaurs and dogs and cats and brains. It's great.
That's very cool.
Speaking of which, of media for children that is like science adjacent, very briefly, do either of you remember the books Stella Luna or Verdi? Because I was talking about them recently. People were looking at me like I'm crazy. Like, those exist, right? Like, they're beautiful. One is about like a green tree python turning from yellow to green. And the other one's like a fruit bat or so. I don't know. They're great.
I don't think I remember. Oh, I do. I've seen this cover before, but I don't know that I ever actually had a copy of the book.
Oh, so good. So good. It's like honestly half of the reason why I grew up being such an animal nerd. I had those the time I was like 2 or 3. So making a little plug for that author.
I had the Time Life books on just like the Time Life dinosaurs, Time Life cats, and those were the ones that I just read until they fell out of their spines over and over again.
Yeah, I can't say I'm familiar with either of those books, and I can't say I read a bunch of animal books when I was a kid. I did spend loads of time outside rolling over logs and collecting salamanders and catching frogs and snakes and going fishing, but I guess I didn't read a lot of animal books. So, I'm not.
That's way better. That's the classic repertoire.
It was good. It was good.
Well, nobody asked, but I'll tell you what I'm drinking.
Sorry.
What are you drinking, Ryan?
It's been chilly, so I wanted to have a darker beer that was a little more refreshing, probably not the right word, but warming in the cold weather. And I thought, we're talking about lizards, so I will have the thing that lizards don't make, which is milk in the form of a milk stout. So I'm having a Black Rock Milk Stout by Devil's Backbone Brewing Company in your neck of the woods, Kelly, Virginia.
Ooh,
Lexington, Virginia,
tasty.
to be specific.
Okay, here's a bad stout opinion. I have a trash palate. I've always found stouts to taste a little bit like soy sauce. Not in an altogether unpleasant way, I guess I'll add, but definitely a little soy saucy.
Have you taken the super taster test?
I haven't. What is that? Actually, not familiar.
So when young human mammals or children or young, I guess is the term, they have an increased sensitivity and density of bitter taste bud receptors on their tongue. And it is thought that that is an evolutionary mechanism because many plants that produce secondary metabolites in the form of alkaloids, which are toxic, alkaloids also taste bitter. So, if for example, cilantro has that bitter flavor because the secondary compound in the form of an alkaloid. And it's thought that, because kids put so many things in their mouth, and that's kind of how they learn about their environment and develop their immune system, that they needed this hypersensitivity to bitterness in order to prevent themselves from wanting to spit out things that would be toxic to them. Much like lactose tolerance, some people retain this trait into adulthood. So they have an increased sensitivity to bitterness. So those are usually the people that can't handle like hoppy beers or can't stand the taste of cilantro because they say it tastes like soap. And there's this one chemical that you can get on like a little strip of paper, and you literally just put it on your tongue. And if you can taste it, you have this condition, you are a super taster. And if you can't taste it, you are not.
That's really interesting. Wow, I'm gonna have to look that up. I'm really curious now because I love bitter stuff and coffee, hoppy beer, etc. But used to hate cilantro. It's growing on me. So I don't know. Ambivert of bitter flavors. Another very brief segue. Speaking of children and poisons, this is like my favorite thing to plug for people too. Is that there's a Facebook group. Literally, the only reason that Facebook is still worth it to me. That's called Poisons Help, which is a group of mycologists and botanists that are on call 24/7, or rather just by volunteer basis, I guess, to identify pictures of things that people's children or pets have eaten. And it's insane how fast they do it, like to tell poison control and accurate ID. So I've learned way more about identification of fungi, plants, etc. just from watching this. You can't comment if you're not, the person whose child is poisoned, obviously, or else it clogs up the works. But it's a fascinating learning group if you just want to read the posts.
What's it called again?
So the
Poisons Help. So a mushroom and plant ID. I can't remember, but it's definitely Poisons Help, something along those lines.
That's really cool.
Mm-hmm.
So the chemical that's in vegetables like Brussels sprouts, kale, broccoli it's a glucosinolate. And the chemical that you can use, you get these little test strips that you can test if you're a super taster, is phenylthiocarbamide, PTC.
Huh. I'm going to order one. I'm curious. Thanks for letting me know.
For sure. You should, the next, COVID safe party you have, everyone can test themselves for super taster.
Oh, hell yeah. I'm going to look so cool. Great.
Well, speaking of having great taste, you also do wildlife hikes and photography. And we're going to talk about that a little bit in our next segment coming up right now. Gina, you are super active on Twitter, and you also like getting people outside, so you use your Twitter account to sort of explore the natural world, and you also are involved in something called Frog Watch. So I just want to talk to you a little bit about your social media, your citizen science, your photography, your nature hikes. I'm excited to talk about all that. So where do you want to start?
Sure. So I can start with Frog Watch, just because honestly, that's a large part of the reason I became active on Twitter in the first place a little over a year ago, because I had just gotten involved with Frog Watch and I was so excited and I wanted people to come with me and et cetera. So Frog Watch is a community or citizen science organization that's affiliated with the AZA, the Association of Zoos and Aquariums. And the point of it is basically to get volunteers from communities across the country out into nature in the evening to monitor frog calling activity. So the basic structure of a Frog Watch trip is you go out after sunset, shortly after sunset, when the frogs are generally becoming active. And the trip leader, which is me, oh my gosh, will tell you what species that we're listening for. So we have like 10 to 12 in the New Orleans area that we usually ask people to listen for. And then I'll play a short recording of the call, just so people can hear what we're actually supposed to be listening for if they're not super familiar with the species. And then we go on this hike through a boardwalk trail through the swamp and stop at a couple of different sites along the trail And then we pause, take a few minutes where all phones are off, everybody's silent, no lights, no sound, no nothing. And at the end of that sort of acclimation period, which is intended to get the frogs used to our presence and just, kind of resuming their normal activity, we will listen for a few minutes and then kind of all confer about what we think we heard, how many we think that we heard of each species. So if it's just a single call, we'll record that. If it's a couple overlapping ones, or if it's a full-blown chorus. And it can be pretty noisy sometimes. So it's hard to tease out all of these individual species if you don't have a lot of experience listening for them. So that's why you have the trip leader to sort of interpret these frog calling things.
So how do the trip leaders get picked in the first place? Like how did you get picked to be a trip leader for this organization? Do you have to pass an exam to show that you know frog calls?
No, I was just the nerdiest participant on one of the hikes. And the person who was running the program for the Jean Lafitte Barataria Preserve at the time was looking for volunteers to make it more frequent because she was running a trip about once a month, I think, and wanted to do them biweekly. So I'm not the only one. There are a couple of us that do these trips, and we've just started getting going again in full force with COVID restrictions in place. But yeah, so anybody can do it. It's sort of an honor system kind of thing. So I take it very seriously, and I think most people who would step up into that position do. But no, there's no test or anything. Just make sure that if you're going to sign up for it, you're willing to put in the work to really familiarize yourself with these calls.
And then do the data get used to answer specific questions, or is it more of a growing database that might get used at some point?
More the latter. So the database has been growing since I think about 1997. So going back pretty far. And primarily what you use these data points for is phenology questions. So phenology meaning patterns of seasonal fluctuations in activity, presence, visibility, etc. So for frogs, phenology studies would probably focus on when they become active in the spring, when they cease activity in the fall, stuff like that, because it can be related to climate change as well. Like temperature cues are primarily what influence frog calling to start and to stop. So really important to keep track of these patterns and the fluctuations in them over time.
So what's the diversity of frogs like down in southern Louisiana? I imagine it's high.
Yes, so there are a lot of frogs that are theoretically present in the park, and then a lot of frogs that, yeah, right? Theoretical frog physics.
That's a band name for sure.
But yeah, there are always the usual suspects that we expect to hear at certain times in the year. And I enjoy having a little mnemonic for each of them, at least to the degree that I can, just because it helps people remember. My very favorite one is for the narrow-mouthed toad, which is called a sheep frog sometimes colloquially, just because it literally sounds like a sheep, a really angry sheep when it's calling. And I'm just going to put myself out there. It's like, meh. So very, very recognizable. But we've also got green tree frogs, gray tree frogs and Cope's gray tree frogs, green and bronze frogs, which are very closely related. The bronze frog is a subspecies of the green. We've got bird voice tree frogs, squirrel tree frogs, Gulf Coast toads, spring peepers. God, I'm just going to keep going. Nobody can stop me. Pig frog, leopard frog, bullfrog. Okay.
I'll butt in to point out something that I noticed when I was studying abroad in Costa Rica, and I was learning a lot of these amphibian and herp and a lot of IDs for the natural world generally. But one thing that stood out to me about frogs and toads in particular is it seemed like there was a not 100% consistent, but a fairly consistent naming convention of the first word is some sort of descriptor, usually about the appearance of the animal. Smoky, green, bronze, right? Mm-hmm. The second word was a descriptor of where it lives: jungle, pond, tree. And then the third word was always just frog or toad. So you got your gouty tree frog, your smoky jungle toad, your and yeah, so it seemed like there was a I don't know why that applies to frogs and toads more than any other group of animals I've come across, but that always kind of made me laugh.
You know what? I'll take it though, because I know that the birding community is having kind of a reckoning about naming birds after historical figures upon whom the light of history does not shine kindly, to say that nicely. So I'm much happier to have kind of the silly, location-based names than people names, because those will stand the test of time.
Yeah.
And so is your frog knowledge, would you say it's hyper local to southern Louisiana, or how could we drop you in a different frog spot around the world and you would be able to start identifying things, or what's sort of the transferability of your skill set?
So my call ID is pretty local. I can figure out how to identify almost anything by call, just with the magic of Google. But my visual ID is pretty good, just because biogeography is something I've always been very interested in. So meaning the distribution of taxa across geographical spaces. And I kind of made a game out of this with fish. In an ichthyology class I took in undergrad, I thought this was fascinating. Part of a test that we had was to tell where we were in the world based on, just vague description of fish traits. So I can sort of do that with frogs too, because there are a lot of different broad groups that you can only find in certain places. So fairly transferable, I would say.
So how does the group work? So like I've gone out on nature walks with people who can't put their phone away, just seem incapable of doing it. Do you ever have problems with people not sort of staying in the spirit of the event, or do you really get like very particular kinds of people who come out and stay on task?
Definitely the latter, kind of trudging into the swamp at night where there are alligators and feral hogs and stuff. Like it's mostly people who are really into it. So the one problem I've had is with a gentleman by whom this is no fault of his own, but I don't think that he could really hear that he was coughing, like, and kind of breathing and shuffling very loudly during these acclimation periods. So yeah, it's not a big deal. The frogs really aren't that sensitive to small noise like that, and I want it to be accessible for everybody. So definitely not a big deal, but I don't run into people who can't put their phones away very frequently. It would make me very sad because, I get excited. I don't want them to spoil the frog listening. So
totally.
Do you have any great like stories of these trips where somebody, accidentally almost stumbled over an alligator or got really excited about a particular kind of frog that they didn't think they would get excited about or something along those lines?
Okay, so this one is not unique to any one trip, but it's my very favorite part of every trip because there's something, just the legendary frog tube that we pass every hike. So at the very end of this trail, it's not a loop, it's just a straight trail out into the swamp and then back. There's a railing that has no caps on either end. So it's just a metal tube and it is full of frogs. 24/7, 365. It is full of little squirrel and green tree frogs. And I can take people and shine a little flashlight into it and they just so it's so cute. But another notable instance, we see a lot of alligator predation late at night. So we'll see them grab a nutria, do the death roll, and we cheer because nutria are terrible. But
yeah, it's an invasive species.
Horrible invasive species, yes. So a lot of cool stuff that you really only do see at night like that.
I really want to go on one of these tours with you.
That sounds amazing.
Come with us.
Yeah, I think that's my next question is, so obviously, you're doing things in a COVID safe way as much as possible. And I imagine that means that you're more or less limited to your New Orleans and surrounding areas community. But for folks who want to get involved, either who happen to be lucky enough to live in the New Orleans area, and I say lucky enough because I love New Orleans, it's one of my favorite cities in the world, or are there other opportunities in other parts of the country for people to be involved with Frog Watch?
So there are tons and tons and tons of local Frog Watch chapters all across the country. Very likely you could throw a rock and hit one.
Please don't hit any frogs.
Yeah, right. Yeah. Oh, save the frogs. They've got enough to worry about. But yeah, so
sure too.
I love having people from out of town when it's safe come and join me on these. But in the meantime, definitely very common type of community science and outreach program that probably is within driving distance of most people.
That's awesome.
Mm-hmm. And you basically just get on a mailing list and then we'll just announce the bi-weekly trips. If you can make it, great. If you can't, join us on the next one. But yeah, pretty laid back.
Legit gonna sign up for the mailing list if I can find one in the DC area.
That sounds awesome.
Oh, you should. I bet that they're great there. Yeah, the more southern states are usually a little bit more active just because there are frog calling activity, or there is frog calling for longer in the year.
DC is lucky because we have Anacostia National Park, which is the nation's only national park unit dedicated entirely to freshwater aquatic plants.
I didn't know that, and I have lots of family in DC. I got to check that out next time it's safe to come up.
Oh, Anacostia is fantastic. Yeah, let me know when you're in town. I'll show you around the one trail. It's not huge, but yes. It is very nice. And you can also kayak around it because it is marshy enough in parts. So
that's awesome.
Do a little hiking, do a little kayaking. It's a wonderful little spot tucked away over on the eastern edge of DC.
Huh. Ooh, I'll definitely take you up on that. So hope you weren't kidding.
It's on the Anacostia River. It's Kenilworth Park and Aquatic Gardens. I got the name wrong, but
I'm writing that down so I can Google it when we're done. I'm excited.
Yeah, it's a cool spot. So what about your other outreach activities? You seem very active on Twitter and other social media. So what made you want to engage with people in that way? And on the whole, we hear about Twitter being a garbage fire, but I often tell people that when the science community isn't having crazy drama, which seems to be often these days, Twitter can actually be a pretty fun place for science outreach. So can you talk a little bit about your experiences there?
Sure. So I initially didn't approach it from a scientific perspective per se. I just do this a lot, going outside and going on hikes just because, especially once the pandemic started, there was like nothing else to do. So I would find myself out alone, like on these hikes, taking pictures and being like, oh, I wish I could show somebody this picture. It's so cool. And just, it started very small with just me posting stuff I thought was cool that I had found that day. And turns out there's a very large community of like-minded individuals who get very excited over, a cool looking lichen or cute little snail or obviously just beautiful, gorgeous frogs that I see everywhere. So I didn't really approach it with intent for it to become any sort of like educational platform, but the more people seem interested, the more I want to sort of branch out into more educational content rather than strictly entertainment based, just nature photography. So I've been trying to include more information about the species that I'm encountering, like include the species name, the Latin name, etc. underneath the post, tell a little bit about the ecology of the species, stuff like that.
Do you have training in photography, or is it something you're sort of picking up along the way?
So a lot of people are simultaneously surprised and delighted to know that most of the photography I post is just with my iPhone, because that's the camera I always have. I do have some training in DSLR photography, but more often than not, the things I find are spontaneous. So the phone is what I have. I'm starting to do a little bit more intentional shoots with, my quote unquote real camera, so I can produce higher fidelity images and take some better videos from further distances, because phone zoom is really trash, but DSLR obviously is much, much better. So as I get more serious about engaging with people on this platform, I'm starting to elevate my photography, but for now it's sort of a mixture of nice big camera stuff and just goofy phone photos.
Cool.
Yeah. Well, if people are interested in following along, your Twitter handle is Gina Goes Outside. And that's on Twitter. Are there other places on the internet that you would, we will link obviously to Frog Watch USA on the show notes for this episode, but there are other places people can connect with you on the internet, or things you would want them to check out, or resources they should know about?
Well, if we're talking about just resources abstractly, even if you don't care about genetics at all, you should just go scroll through the NCBI database because it's cool. But because I'm a masochist, I actually only use Twitter really for social media. So that is pretty much the only place to find me. But
if only there was a social media platform that was dedicated to sharing nice photos.
Right? Oh my God. Okay.
There isn't. So
I wrestle with it. I wrestle with Instagram. I find it almost like non-functional with all the updates that they've been pushing. I use it sometimes. I keep saying to people that I'm going to be better about it, but I'm not. I'm a liar. Don't believe me. So I'm going to try. I'm going to say it again here. I'll hold myself accountable. I will try to be better about posting stuff on Instagram.
I guess my last question has to be favorite frog and favorite other herp, non-frog herp.
Oh my God, favorite frog. How would you ask me that?
I always want to end on a really hard-hitting question. This is real journalism, folks.
Mm-hmm. Okay, well, I love the Wallace's flying frog. They're also called parachute frogs, and I'm not sure if either of you have ever seen them, but they have these absolutely fascinating feet that have really dramatically webbed toes, and they can use these webs to sort of catch a little bit of air. So they're not flying frogs, actually, but they're gliding frogs, and they're really cool, and they're really cute. They're from Southeast Asia, so maybe I'll get to see one in the wild someday. But Wallace's flying frog, just the pinnacle of what a frog could be. Strawberry poison frogs are also extremely cute and good, very phenotypically variable, which I think is neat.
Is it Wallace's flying frogs because of Alfred Russel Wallace?
Yes, it is.
Oh, cool.
The Darwin of Southeast Asia?
Yes, it is. Yeah, good call. But yeah.
They're so cute.
They're I'm looking at a picture of one right now. I'm just enraptured.
Just because they're gliding, like that's still a type of flight. It's just non-powered flight. They're not flapping their little frog feet. They're just gliding on them. God,
that's a great mental image, though.
How is there not a superhero that is this frog?
Be the change you want to see. It's never too late.
Right? Frogman.
Oh man, non-frog herp. I love snakes. That's actually my origin story. I have several pet snakes, and I have for my whole life. I think I was like a year old when my dad got me my first pet corn snake, which just passed away like a year ago. It was old as dirt. So yeah, I love snakes. I think the Mangshan Mountain pit viper is my favorite snake, probably. It's absolutely gorgeous. If y'all have time to look up a picture. They're also interesting because they're actually unique among vipers in that they lay eggs. So most vipers actually have live birth. So I think that's cool. Maybe not everybody does, but
no, that's super cool. I think people I think people are surprised by the diversity of, because like there's lots of fish that give live birth. Like live birth is not a strictly speaking mammalian trait. There's lots of other animals that have figured out how to do a version of it. A lot of sharks give live birth. So yeah, it is very cool that I have a particular soft spot for parental care in reptiles. So like, I think my favorite reptiles tend to be the ones that like look after their little baby reptiles. So,
Oh,
like
crocodilians.
the big mama crocodile,
Yes.
like protecting all her little babies with them like sitting in her mouth and just keeping all the nasty teenage crocodiles away from them. And yeah, it's
When the charming Suriname toads.
Oh, okay. This is such a controversial opinion in my household because I love them. I love it. Like, I want to go to Suriname just to see one.
They're awesome.
We have them at our local zoo. Yeah. And my fiance hates me when we go there because I want to sit there in front of him and he thinks they're so gross. I love them. I totally agree. So yeah. Toad Defense Force.
Toad Defense Force. What are your thoughts? This is a completely random question, and I know we're a little over time, so I'll let you go here in a second. What are your thoughts on the fact that we call herpetology the study of amphibians and reptiles when that is not a monophyletic group? Shouldn't there be? And if we were treating it as such, shouldn't it also include birds, which are technically a highly derived archosaur?
No, you're totally right. I like it because it's convenient to me specifically, you know?
That's fair.
So it's fine. You are definitely right. I think that going into that argument, like you could take that down so many different pathways with like paraphyles and, you know, whatever else.
Polyphyles.
Yeah. There's so much. Like nothing's real. Like I also like it because it sounds funny. So this is a very unscientific opinion of mine, but for personal reasons, I will not be objecting to the word herpetology.
Scientists have made worse calls for worse reasons, so
Exactly.
I think you're okay.
Amen.
Well, thank you so much for joining us. The correct answer was hellbender for the best herp. So
Okay. I can definitely put that on my like list of honor. They're so cute. I used to live. I'm so sorry. I have to say this. I used to live in Pennsylvania, and when I was a little kid, because that's where you can find them actually in the wild. I'd spent hours and hours and hours, like every day one summer, looking for a hellbender, and I never did. It's the white whale, you know. But,
I mean,
someday,
they're pretty well camouflaged, and they live under rocks most of the time.
Yeah, I know that bastard was just like watching me, mocking me. Someday.
When I talk about the time I've spent in the tropical rainforests of Central America, people often ask if you've seen certain animals. So like I have encountered a fertilance, which is this really venomous snake.
Oh my god! Yeah, love them.
Really cool. They're related. They're like distantly related to rattlesnakes, and they will actually tap their tail against a leaf or something nearby to like simulate a rattle noise. Like it's really cool that they have this like shared behavior, even though they don't have the actual rattle. But so I've encountered these really amazing animals, but a lot of times people want to know the big megafauna, the jaguars, the mountain lions, right? And my response, and I believe this is true, and I have no way to verify it, but I believe this is true, is that I've never seen a jaguar when I've been in the rainforest, but I'm pretty sure they've seen me. Like if they're anywhere nearby, they know that I'm there. They can easily keep an eye on me without me ever noticing them if they want to. And I think that should give you pause.
So leaving on a note that will just strike terror into the hearts of people who are enthusiastic about tropical habitats everywhere. I'm recalling a story on that note of a researcher who had peed on a tree while doing field work once, just totally unrelated field work. And then apparently, I think it was a jaguar. I think it was like Central South America, and it followed her for weeks. Like she just pissed it off, like peed on the tree, and she would see it like out of the corner of her eye, stalking her for like weeks at a time, which is my nightmare.
That is
I don't mess
scary.
with mammals.
Yeah.
That's why you got to do the thing that the cacao harvesters in Southeast Asia do, is you put the plastic Halloween mask on the back of your head.
Oh my God, that's genius.
Because cats, they like to attack from behind. So if they see eyes looking back at them, they think they need to get around the other side of you. And if they get around the other side and there's still a pair of eyes looking at them, they give up.
And they don't figure out that the eyes aren't blinking?
Man, do all podcast hosts have such good factoids?
They did a study recently. We didn't talk about this on the show, Kelly, which may be one of the greatest oversights in science podcasting history. Uh-huh. There was a study that just came out last year, I want to say, where they did an experiment in India with cattle, where they had a control group of regular cattle and then a group of cattle where they had painted eyeballs on their butts. And that the experimental group got attacked by tigers far less often than the control group because the tigers saw eyes and just didn't bother.
Ryan, it's not too late.
That's so funny. I really want this to become like a widespread agricultural practice.
May I please send you I'm going to paste a link in the chat here on our recording software. Just click on that and enjoy.
No, the indignity.
We've got to address this oversight, Ryan.
Okay.
Those are pretty good eyes.
Right? There's like some artistic merit to what they were doing too, which I think is all the more impressive, but it worked.
That's so funny. Oh my God.
They did not get attacked as often. So that is the trick. If you want to do field work in an area where there are big cats and you're worried about getting stalked, is Halloween mask worn on the back of your head.
And one on my butt for a good measure. That's what I'm getting from that.
No, you got to paint there. You can't use a mask.
Yeah.
I feel like this is a strategy where if you overdo it, they're going to figure it out, though. Like if there's, butt eyes everywhere, eventually, don't you think they'll figure out that like the butt eyes that don't blink,
You're
can
worried
go
about
after
red
those?
queening, hypothesizing our own eye butts?
I am, yeah.
But Kelly, you're ruining our potential new yoga pant business. Anti-tiger yoga pants. We could give part of the proceeds.
Never mind.
We could give part of the proceeds to tiger conservation. Guys, this is such a win-win.
Oh my God, $1 million idea.
Copyright, Science Sort of 2021. You can't take this idea out. Well, thank you so much for joining us, Gina. This was a ton of fun. We loved having you. And I hope that as you get results for this really amazing project that you're doing, that you will come back on the show and tell us more about it. And you know what, Kelly?
What?
We've done field trip shows before. I think when it's COVID safe, we got to go do a frog watch trip with Gina and record it.
Hell yeah.
One of us. One of us. Yes. Thank you so much for having me. It has really been so much fun. I'll let you know also when I come back how the yoga pants idea is faring in the market.
No, that's our joint venture. How dare you?
Yeah.
Don't steal that.
All right, cool. Patent's in the mail.
I'm going to mail it to myself. That's legally binding. All right. Well, Gina Zwicky on Twitter, twitter.com/Gina Goes Outside. A great follow if you are on that particular social media platform. And until next time, thanks again for joining us, Gina, and have a great rest of your day.
Thanks, Gina.
Thank you. You as well.
All right. So that was our interview with Gina Zwicky. Thanks again to her for giving us some of her evening to talk about all the cool herp stuff she's doing. Love herps.
She's super awesome.
Yeah. I've been excited about having her on the show for a little while. I'm glad it finally worked out. You know who else is awesome, Kelly?
The people who listen to Science Sort of.
That's correct.
The PaleoPOWs.
We they're the Paleo Posse. We are the PaleoPOWs. We've been over this so many times. I don't have time
so many times. That's right.
But
every time I'm on, I think we go over this. Actually,
it's not just you; it's everyone. It's the early days of our branding efforts were somewhat confused and remain confused to this day. But the one thing we're not confused about is how awesome our listeners are, and they're so awesome, in fact, that we dedicate our entire final segment to them, and that segment is called the PaleoPOW. Final segment of the show where we delight in feedback from you, our beloved listeners. Do you want to go first or second, Kelly? Dealer's choice here.
I'll go first.
Take it away.
So, all right. So we want to give a very heartfelt thanks to Amanda B for her recurring PayPal donation. Thank you so much, Amanda. We really appreciate it.
And what's cool about Amanda is this is actually the second time she's signed up for a PayPal recurring donation. So we've thanked Amanda before. So if you're thinking to yourself, now I remember in episode 226, they thanked Amanda B before. This feels like a scam. Not a scam. The recurring donation either lapsed or sometimes when you get a new credit card, it automatically cancels and you have to reset it up. And not everyone does. Amanda B did. So that's why we want to thank her again.
Way to go, Amanda B.
Yeah, and obviously if you would like to set up a recurring donation, we don't have it set up to do that way on PayPal anymore. You can give us a one-time donation on PayPal if that's more your jam. If you would like a recurring situation for yourself, you can do that via Patreon at patreon.com/sciencesortof. We have multiple different levels. You can also input a custom level if you know exactly how much you would prefer to give, because we came up with silly science-based numbers that might confuse your accountant. Or not. Maybe you've got an accountant who's a fan of science and they're going to be really excited. So you get some cool rewards over there on the Patreon if you choose to sign up in that way. And one of our patrons is actually responsible for our second PaleoPOW of the evening, and that is, I'm not sure how to pronounce their Twitter handle, but it's Z_DEIGE. Hopefully They don't mind me sharing their Twitter handle publicly, but they sent a public tweet to us, so I think it's fair game. And what they said was, just listen to the latest episode, and this is referring to episode 333. I think Tim did a really good job with the interviews, smiley face. I also really like the dust storm segment. I never even would have considered that dust storms could be an important part of ecosystems by delivering nutrients, iron, etc. Shocked face. So that was a really great tweet, because for folks who've haven't yet listened to episode 333, that was sort of, we tend to do an episode focused on some of the research coming out of the American Geophysical Union annual meeting every year. That meeting was virtual last year, and I was busy with some professional commitments at that meeting, and so I didn't really have the mental bandwidth. I had the internet bandwidth, but I did not have the mental bandwidth to deal with doing a bunch of interviews like I normally would. And our very own audio producer, Tim Dobbs, stepped up and did interviews for me and edited the interviews. And then we did like a almost Radio Lab style, where I'm the host and Tim comes on and says, hey, let me tell you the story about what I went out in the world and recorded for you. And I got to sit there and just revel in it. It was a delight. And I was so proud of Tim. And it was just like the little baby bird taking off out of the nest, and it just made my heart swell. And I'm so glad people enjoyed that episode, because
go Tim.
I was so tickled that it worked out and that Tim was willing to do it for us. And just very pleased as punch, as they say.
Tim's a good dude.
Yeah, so I hope other folks are enjoying that episode. And if you are enjoying that episode, give us more feedback so we can tell Tim he's doing a good job. The only other thing I would like to cover before I let you go, Kelly, is, are you familiar with bookshop.org?
Tell me about bookshop.org, Ryan.
Bookshop.org is a place online you can buy books. Now, this may be a new concept for a lot of folks. Turns out people like buying books on the internet, and there are several places online that have made it very easy to do that. Not all of those places are super great to their employees or to the publishers and the authors that write the books. However, bookshop.com is different. This is not an ad. I don't know why I'm doing this. No, to be perfectly frank, bookshop.org is a site that was built to help local bookstores fulfill online orders to huge, internet-wide audiences. So basically, if you want to buy a book, you search for it on bookshop.org, and then when you go to check out, it connects you to a local bookshop in your area, so the money you're spending is going to both support the author and support a local bookshop in your area, and you're still getting to order your books with the convenience of online shopping. They have an affiliate program, and we are an affiliate. So if you go to bookshop.org/shop/science sort of, any book you buy, in addition to supporting a local bookstore, will also support our podcast.
Win.
Yeah, we're really excited about it. Our shop page gives us the option to create lists. So we have lists that I'm continuing to populate. So we've had a lot of authors on the show over the years. So I'm trying to get their books all on a list. But Kelly, you've actually written a book.
The book is Soonish, 10 Emerging Technologies That'll Improve and or Ruin Everything.
And when you go to bookshop.org/shop/science sort of, you will see the first list is books by us, and it is comprised of one book. But maybe more down the line. But I just wanted folks to know that if they haven't yet picked up a copy of Soonish, they can and should consider doing that on our shop at bookshop.org.
Thanks, Ryan.
Absolutely. You got anything to plug, Kelly? How's things? How's life?
Oh, life's good. We're working on book number 2, but it'll be a couple years before we can add that to bookshop.org, to our list on there. So working on that mostly, and parenting in a pandemic, and running some research projects. What about you?
Also running some research projects, working at my new job, which I guess I haven't really talked about on the show, but we're running long, so I'm not going to get into that now. But I got a new job, which I'm excited about, and may have some opportunities to partner with folks who listen to the show, so stay tuned for that. I think in a more acute way, I got to do the iFanboy Special Edition show on the new Batman animated film called Batman Soul of the Dragon, which is a full-on animated 70s style kung fu film. Batman is practically a side character, and it was a ton of fun. So if folks like kung fu movies, and if they are into the idea that everybody really was kung fu fighting, I would recommend going and watching that movie and then checking out the podcast where we discuss that movie in detail.
Nice. Oh, and I forgot to mention, I was a guest on a podcast. I was on Daniel and Jorge Explain the Universe. Yeah, Daniel and I did an episode on the science behind whether or not we can make invisibility cloaks. And, it's funny, if you had told me a decade ago that one day I would be filling in for the PhD comics guy, it would have blown my mind. I don't really read comics these days, but that was like my favorite comic in the whole entire world. And anyway, it was super cool to be on the episode. And Daniel, the host that I was on the show with, is a great guy, and we had a fun talk about invisibility cloaks. So you should check that out.
I don't have anything that cool to plug, so I'll just say I've been getting back into archery as a pandemic activity, and that's been fun too.
Oh, nice. That's awesome.
Yeah.
I've been learning Russian.
Whoa. All right. That's cool.
Yeah, I've gotten into the history of the space race. And if you're going to read about the space race, it's helpful to speak some Russian. So I've decided I was spending too much time on Facebook and Twitter, and it was making me depressed. So now whenever I want to go on Facebook and Twitter, instead, I pull out Duolingo or my Russian workbooks, and I do some Russian instead. And that's keeping me sane, actually.
I started the Russian course on Duolingo, and when I got maybe 10 minutes in, and it said, okay, now plug in your Cyrillic keyboard, I was like, I'm out. Sorry. Can't do it. Can't.
Oh, you don't have to plug it in. You just, well, so I've been doing it on my iPhone, and I just load the Cyrillic keyboard, and it just pops up instead of my usual keyboard. And there's a little button in the corner now, and it switches from Cyrillic to English.
I love languages. I love learning new languages. I still find a hurdle that I have yet to mentally overcome is learning a language that requires a new alphabet, which I know is, whatever centric of me, but it's just, that's my particular mental hang up that I haven't yet figured out a way around overcoming.
This one's kind of killing me because it's got a lot of the same letters that we have, but they're different. Like what should be P is R. And so it's like, I'm having trouble catching up. C is an S, the S sound, I think. And so that the fact that it's the same letter, but a different sound is really throwing me, but I've been sticking with it so far.
Cool. That's exciting. Well, the next time we have a Russian parasitologist on, which is weird, that's a thing that's already happened. You guys can,
yeah,
we can have a little Russian conversation.
That would be good. I'm down.
All right. Well, thank you, Kelly, for joining me. This was a lot of fun. I had a lot of fun chatting with you and Gina this evening.
Yeah, thank you. I had a lot of fun. I'm glad I was able to come back on. I've missed being on the show, but of course, yeah, right. Life is crazy.
I started corresponding with Gina, and I'm just like, this is a woman who's excited about taking photos of various herps and other animals in her environment. Who else do I know who's into that lately? Better loop in the old
well, and she's into parasites.
Right. So
yeah, we're kindred spirits, I think.
Yeah, so I was happy to make this love connection.
I appreciate it.
All right, well, scienceorder.com for the show notes for this episode with all the links you need to check out the stuff that we talked about. Thank you for listening. Come back next time. I can tell you, do you want a sneak preview of what the topic's going to be? Since I already have the recording lined up, I just need to do it.
Yeah, tell me.
Yeast. We're going back to yeast. We've talked about yeast before. Get ready for some yeast updates.
It's the gift that keeps on giving.
An excellent fungus that we can all enjoy. So with that, I just want to make sure we thank Rob Heath for audio production for this episode. Thank you for listening. Be sure to come back next time where you'll get a whole lot more yeasty science. Sort of. And we forgot to have Gina say sort of, which is something I'm trying to remember to have the guest to do. Sometimes you just forget things, Kelly.
There's a lot on people's minds these days.
There sure is.
Visit sciencesortof.com for show notes, links to all the stories we talked about, and ways to interact with the hosts, guests, and other listeners. Science Sort of is brought to you by the Brachiolobe Media Network of podcasts. That's all for this week. See you next time on Science Sort of.
Everybody warm? Seems like that's the other major issue across the U.S. today.
I've been wrapped up like a little panda bear all day long. Like I look like I'm wearing like a trash bag. I have like a double, triple XL coat on right now with like 4 layers underneath it in my home. So that being said,
is your power out or?
No, I'm just a little baby. The houses down here are really poorly insulated.
Oh yeah, fair enough.
If your power was out and you were still hopping on the podcast with us,
yeah, right. We're used to stuff like that. Yeah.
You know what? We can reschedule.
We're good.